Horizontal, again, is passing something to your neighbor, right? Across genetics, so to speak. We call that, guys, horizontal, all right? Don't worry about recombinant. No vertical gene transfer. And I'm going to talk about this process of genetic recombination, not the word recombinant, genetic recombination. You guys already know this. I don't know why we don't put up on the slide, guys, crossing over, right? But that's exactly what this is. Does anybody remember what that is? Crossing over? Somebody just spoke for everybody and said, no, nobody remembers. Okay?
She's right.
You do, yes. Damn, okay. The other answer. Can you explain it?
So when you're chromosomes the legs to touch, and so like
they literally cross over, right? Yeah. When you get ready to divide, guys, when you're going through mitosis or meiosis, right? You got to duplicate your chromosomes. You got to line them up, as Katie said, and then you end up, guys, forming this structure called a tetrad. I don't care, but you see how we have 4 there? That's what tetrad means. Tetra means 4. All I want you guys to know is a reminder about how we swap. The subtle cue, fold the arms, I turn off the fan. Is that when 4 chromatids come together, sister chromatids, chromosomes, doesn't matter, maybe chromatid, jerks a little bit in your memory, okay, doesn't matter. When we've got 4 chromosomes laying next to each other like this, there's a thing that happens. Kind of like when you've got the 2 transfer RNAs and the amino acids are next to each other, a peptide bond just forms. You lay 4 pieces of DNA next to each other, right? Mom's set and dad's set has been duplicated, but you don't have to worry about that. Just remember 4. When you see that, the inner pieces, not the outer pieces, guys, the inner pieces swap. It would have been nice to make these different colors than purple and slightly lighter purple than that color, to really see what you end up with after you swap the genes. Okay? Every time a cell gets ready to divide, they line their stuff up, and their inner chromosomes that we see up here, guys, cross over. That's important. That's why every time your mom and dad have a baby, all you guys look different. Okay? You all look different because every sperm cell we make, every egg cell we ever make is always a little bit different than the other, because right before we make it, we swap the chromosomes. We put the chocolate in the peanut butter, guys, every single time we make a new sex cell. That's why all the eggs are different. That's why all the sperm are different. And the only way you're going to be the same is that if you're using, duplicate sperm or duplicate egg, or one does both, the ways to make twins. Okay? Has to do with, the same genome or 2 copies of the same genome. This is crossing over. We call it homologous recombination, right? Genetic recombination. Just swapping pieces. I was red, mom was blue. We swap pieces. Now I'm red-blue. I'm not red, I'm not blue. I'm red-blue. Okay? That's why you guys kind of look like your dad and kind of look like your mom. Okay? Because you're not red, you're not blue, you're red-blue. A little bit of both. Life loves this. Back a long time ago when this first popped off, life was like, oh, that's a good accident. That's weird. Wish you wouldn't have done that for real, made a mess. But this is great. Why? Because when your genetics, guys, are more jumbled up and more mixed, the more mixed up your genes are, this is a really, really causal relationship now. If you're mixed, you're healthier. That's simple. If you're mixed, you're healthier. German Shepherd, mutt from the Humane Society, that mutt on average, guys, is going to be smarter. Right? Maybe we talked about this, guys, right? We've studied this. Fish swim faster. Rats find the cheese faster. They're smarter with the maze. They can run the wheel longer. Birds can diving speeds faster. Birds can hit the mouse quicker. Everything. Mouse can duck from the bird. Everything you can imagine, guys. Always when you're mixed, you win. Okay? That's a real big deal. That's a real big deal because that explains why this even happens. This is a really weird accident. Why do we do this? Because when this happens, you end up being mixed, right? This DNA polymerase thing. It's actually good that the thing screws up. We would have fixed this A. We would have fired DNA polymerase a long time ago with his job performance like that. Okay, if he didn't provide this accidental, really awesome result. Okay, a lot of things happen in science like that. Cooking happens like that. Burnt this. Ooh, it's great. Do that again. Okay. All right. So mixing is outstanding, guys. This is a way to do it. Why do we have sex for real? We could just bud. We could just make babies off of our arms, right? It's probably less complicated, right, ladies? Yeah? That'd be nice sometimes, right? Okay. Bud, get out. Okay? Is that my babies are a mix of your sperm and a mix of my egg. That's why. You know, we got single-celled organisms. We already know this, guys. We got all kinds of crap that's haploid out here. Doesn't mean you're unsuccessful. It means you're going to win more often if you're mixed. If I can take sperm DNA and jumble it up with this other egg DNA and make these weird combinations, everybody's better than if you just clone yourself over and over. Better. Okay? It's not, you know, it's like C passes, but an A is better. That kind of thing. All right? Cloning is like C genetics, but it still works. Okay? All right. So we got to learn some fancy genetic mixing. Okay? Two of these, transformation and transduction, we already kind of know about bacterial conjugation. Again, hook up the pilus, pass me some plasmids, okay? I'm cool, I'm resistant now. That poison doesn't kill me because you gave me the secret DNA. All right, but these two require just a tad bit of brain power, guys. All right, and these are two examples of horizontal gene transfer, okay? Here we go. The first one is an extremely important experiment. Talk about classic, guys. It really doesn't get much more classic than this experiment right here. This happened in 1942, guys. I get it wrong, 42, 44, something like that, where it's very, very important for two reasons, this experiment with these mice. Okay, this is Streptococcus pneumoniae. This is the one that we looked at, guys, the other night, that when we put it on blood agar. This was the dude in the middle that was olive green that had partial lysis. I told you guys the one that gives us ear infection and nasal infection, that's what this is. But it kills mice, okay? It's called pneumonia for a reason, by the way, okay? It causes pneumonia. But it's important for two reasons. Number one, it identifies this wacko bacteria mechanism. This idea that I can just go plug up to Trey here, and again, he needs some, you know, hey man, I gotta go do this little presentation for my kids on biology. Give me some, you know, I need some genes real quick. Okay, you know, he's go over there, and now he's like, you know, master's level biologist cat breaking it down for his son and daughter. You know, this is my dad, right? He just passed the DNA right to him. I usually pass DNA down to my kids. Here I just did it across the room. Okay, we've talked about that with the conjugation thing, but guys, this is the first time that it's ever happened, like a bacterium went over and said, not really passing it, like from one to the other with a pilus, like, whoa, hey, I just found some DNA. I could use that, right? Just picked it up off the sidewalk, like a cap off a soda bottle. Yeah. Okay? That was way inside. That's for one person, I think two. Miss Allen was there too. Okay? Then this is craziness, and we got to know why it's so bad too. It's crazy, first of all, but it's bad. It's real bad. Okay? It's bad for us today. And then, so that's the first thing. We identified this thing called transformation. That these bacteria can, like Trey, can transform into a master's level biologist by hooking up to a dude that's got the master's biology genes. Okay? That is weird, guys. I mean transform into me. That's, okay, all right, I didn't have to do that to you, but anyway, let's talk about the second thing that it did. This experiment, guys, documented the fact that DNA is the genetic material. There was a lot of debate, guys, in the 40s, a lot of debate whether protein or DNA was the genetic material, the stuff that gave us all of our traits. And because protein can do almost anything, there was a lot of people thinking protein. You guys ever heard of Linus Pauling? Okay, Linus Pauling was one of the protein cats. He was the one who was guarantee, I know it's protein. Okay? So anyway, two things. Evidence that DNA is the genetic material. Okay, that's what this experiment did. And number two, documented for the first time this wacko process called transformation. The idea that a bacterium can be laying there on the handle of the sink in your clinical setting, right, in your patient's room, I've got strep right there, right? Normal, staph, normal staph, and then there's MRSA right next to it. Okay, right, like here on the pipe is regular old staph all over the place everywhere, and then here's MRSA, because we're in a hospital, and MRSA's all over every hospital. So, a little bit of water, right? Now these guys are interacting. Now what does this guy do to this guy? He passes him the MRSA genes, okay? And now this guy was regular old staph infection, now he's MRSA. Because we passed that gene. This guy transformed into MRSA because he sucked up DNA from his environment. Right in the water on that handle, right that fast. Which is the push, guys, of why we have to know how to clean. Why we really got to understand what bleach is and what soap is and how we clean, you know, what's the temperature of the room, how long do I have to let it set. It all matters because you want zero. This experiment showed us the bad, that you cannot have any, like in your body, out in the clinical environment, everybody's got to be dead. You have to have zero or close to it as humanly possible, because if you let them exist, then they can transform. Anything can happen. We identify the fact they can suck up DNA. Let's say, guys, that you went in and just blew out the room, right? But you still have bacterial DNA in the room, right? Staph can go pick up that DNA, and now it can theoretically transform into something else. Oh, that's bad. But if you're clean, if you take your medication throughout the entire period as it's written on the bottle, you get as close to zero as you possibly can, and you minimize the chances of problems. All right. So let's take this one slow, okay? All right, strep pneumonia. This is the dude that gives us ear infections. All right. It comes in two different strains, everybody. S for smooth and R for rough. And the thing about the smooth and the rough is the colony. When you have a capsule, we know what that is, okay? When you have a capsule, you end up being very, in all of your other bacteria buddies that make a colony, all of y'all have a capsule, you end up, guys, with a colony that's very smooth and silky and shiny looking, all right? But if you don't have a capsule, the colonies that grow look like cream of wheat, okay? Like, you know, grits, okay? But kind of a finer grain than grits. Okay, just like little bumpy colonies. They have a rough contour to them. Okay, so smooth's got the capsule, and rough doesn't have the capsule, S and R. Okay, there's some exam questions right there. All right, there's R and S, and can you keep it straight? Why is it smooth, capsule? Okay, all right. The capsulated form is the lethal form. That's the bad guy. If you have a capsule, you're going to kill. So what Griffith is the dude's name. I'd like you guys to honestly know his name, okay? Griffith, Andy, oops, sorry, old head reference. Yep, God darn it. You guys have heard of Seinfeld, yeah? Okay, good. Just checking. I want to make my age jokes, or my age comments, they're not funny. I take a live capsulated strep and I inject it in the mouse dies, okay? That's observation number one. And then I take this capsulated form and I kill it. I go put it in the heater, I heat it so hot that it kills the cells. But guys, a capsule, this is key, okay, to this story. The capsule is kind of like an eggshell. So I've got the egg and the embryo developing inside, right? That's the living thing. The capsule is just a shell. When I heat treat, I kill the cells, but I still have a shell full of molecules from these guys, okay? I got a shell, got a capsule full of strep goo. That's a bunch of molecules because we killed the cell, all right? Everybody with me? I've got DNA in there. I've got proteins, all kinds of molecules from those cells. Cool. When I take that thing, inject it in the mouse lives. It's just a capsule, right? There's nothing alive to kill you. That makes sense. So far, so good. All right, the third thing. Just here we enter strain R. Just FYI, strain R is not lethal. You take the non-encapsulated form, stick it in the mouse, and the mouse doesn't die. Okay, now I know it's late, this is like a new experiment, but let's kind of slow down and think about for a second. Why do you think the capsule kills and the non-encapsulated form doesn't? What do you know about a capsule? Can't get to it, that's right. Capsule. When we learned about capsule, guys, didn't it say one of the blurbs that may aid in host recognition, may hurt